Practical problem is actually getting to see life elsewhere, if we can find life elsewhere. But there's also the theoretical problem. To be able to find extraterrestrial life, you must be able to recognize it, identify it as life, delineate its properties. And we had a little taste yesterday of trying to get people to agree about a definition of life. But in order to recognize life, we have to be able to define it. But then we have to be able to deprovincialize the concept of life, we have to identify life elsewhere, so you get a vicious circle. So to try to get around some of the problems, the practical problems and the theoretical problems, we can take an observational approach, which overcomes a practical problem through studying biospheres by remote sensing, and overcomes a theoretical problem by prioritizing empirical evidence and furnishing an observational basis that any theoretical approach must explain. The practical and theoretical problems of non-observational theoretical biology are replaced by other problems distinctive to an observational approach. So the observational possibilities are, we're right on the cusp of the technology. We'll soon be able to do exoplanet atmospheric spectroscopy, and we'll be able to contextualize observations of planetary chemospheres on the cosmological timescale, compare the development, and have a sufficient number of comparisons to make a predictive taxonomy of chemospheric development. Some of the hurdles are, we're not quite there with the technology yet, so this is still speculative of what we'll be able to achieve with exoplanet atmospheric spectroscopy. Planetary science and astrobiology don't yet have the conceptual framework to make meaningful use of all the observations we'll eventually be able to make with that technology. Origins of life research is often fragmented by disciplinary boundaries, so there's not the communication there needs to be between geology, biology, and physics and related sciences. And there's no overarching research program to fill in the gaps, presumably the gaps of the gap map that we're going to talk about next on a cosmological scale. So we do have a conceptual framework to categorize stars in a familiar way, the familiar stellar classifications, where we have the Morgan-Keenan on the left that's based on the surface temperature of stars, then a little bit of refinement is the middle number, and then the position of its evolution of the stars, the last Roman numeral. So when we talk about our sun, we know it's a spectral class G2V. But when we come to planetary classifications, this is a non-exhaustive list of some planetary classifications I've happened to come across. This is basically no better than a folk planetology. It's also geocentric and anthropocentric, as we classify planets as things as like super Earths or hot Jupiters, because that's what's familiar to us from our own solar system. So when it comes to Earth, we don't really have a taxonomy that will allow us to say something about, what kind of planet this is in the most generic way. And we need a generic taxonomy so we can put things in the same boxes. And we don't need just any taxonomy. We need a taxonomy specific to astrobiology. We have a geophysical definition of life, a geophysical definition of a planet. And then to that you get there's also an astronomer's definition of a planet, which includes dynamical considerations like whether that planet clears the other orbiting bodies in its gravitational area of influence. We have planetary taxonomies based on size. When we call something an Earth twin or a super Earth or a mega Earth, we're basing it on size. Taxonomies based on composition, like the rough in the previous slide where I showed terrestrial, gaseous, and other kind of planets. The terrestrial planets are believed to form within the frost line, and the gas giants are believed to form outside the frost line. We have some proposed taxonomies based on entire planetary systems that's come out since the Kepler data has come out, and we've been able to reconstruct planetary systems elsewhere. But we require, as I said, a planetary taxonomy specific to origins of life research. So we want to know things like, is the planet geologically active? Does it have plate tectonics? Is there a geodynamo in it that's going to create the geomagnetic field? There's actually been some papers that have written that volcanism can be determined by remote sensing. We want to know if there's energy flows and what kind of energy flows are on the planet. So we want to know about the star, the orbital radius, the Milankovitch cycles, and radioactive abundances in the crust. And once again, the geodynamo that overlaps with geological activity of the planet. And we want to know about the chemical and mineral abundances, obviously. Spectroscopy may eventually allow us to reconstruct a lot of these things over interstellar distances using the full spectrum of techniques that will become available to us by the technology and development now. But we have a nearby test case, an example of Venus, which was discussed yesterday, because we have this, there was a really big deal when there was an announcement of discovery of phosphine in the cloud decks of Venus, and whether that was a biosignature gas. Obviously, we know that the surface of Venus is a very hostile place to life, but there could be life in the cloud decks. But everything about the discovery was controversial. There many papers have been written, some of them about whether there was actually a phosphine detection, how much there was, and whether phosphine is a legitimate biosignature gas. And with Venus, with Mars, we might actually be able to go to Mars and do core samples and do the kind of geology we've done on Earth to reconstruct Earth history. But with Venus, it's not likely that anytime soon we're going to be able to do that kind of reconstructive geology to understand the history of the planet. So biosignatures can be very ambiguous, and there's been a lot written about this too. There's not just one gas, you may need a suite of several different gases present. So a recent paper just came out this year called The Futility of Exoplanet Biosignatures, which is a very despairing conclusion, which the author basically said that biosignature detection isn't going to tell us very much, and the people who are suggesting it don't even have a hypothesis that they want to test. And they suggest instead of this that we should focus on studying life on Earth, finding reachable life in the solar system, making life in the lab, and observing intelligent life via technosignatures. So we need to constrain the ambiguity, and our own Martin Dominic has written, understanding arises with the ability to place things into context, while unique objects will always remain enigmatic forever. And Martin also said there's no roadmap of exploration. Our knowledge needs to be composed from a large number of puzzle stones, some of which have not yet been identified. And here are some of the pieces of the puzzle that Martin shows in his paper. So, while I don't disagree with that, I think we certainly can have at least a partial roadmap, and I want to offer a roadmap for the reconstruction of planetary life histories that goes from an initial planetary taxonomy to the identification of planets of the same taxon, the estimate of ages of the planets in a given taxon, the reconstruction of stages of development, and the taxonomy of planetary chemospheres. So initial planetary taxon, we sort of started on that, as I indicated with an earlier slide. We have these folk planetology category, but that needs to be made systematic. And like we've sort of started on that with the Kepler. This is the Kepler telescope, which gave us an enormous amount of information about exoplanetary systems. You can see a small slice of the galaxy that it focuses on. We need to identify planets of the same taxon in distinct planetary systems, which has also started here. So if you look through the four color dots here, we can identify the same kind of Earth-sized planet in various parts of the sky. And we need to estimate the age of each planet in a given taxon. That's something that we can partially do, but we don't yet have the precision we need in terms of the Hertzsprung-Russell diagram and estimating the age of the star. So we need to be able to do that more precisely so we can get the age of the star, can in some cases give us the age of the planet. And then we need to arrange the planetary stage development based on age and a serial order. So this is a technique that's very well known in archaeology called seriation, and the picture is one of the most famous examples of the decorations on headstones in a New England churchyard. So before computers, archaeologists would actually cut out little strips of paper and they would arrange them in these orders you can see to try to figure out, because people would often move around and they would make pottery in one location and move to another location, but if you track the style of the pottery, you could figure out who went where. And so this was often used before radiometric dating techniques were available to be able to date prehistoric peoples. I'm not getting it to advance here. I've gone several ahead here. Okay, so we do this with stellar evolution in a way. And here's another definition of seriation that's not specific to archaeology. None of us live long enough to see the evolution of a single star, and no scientific research program lasts long enough to see a single star. So how do we understand stellar evolution? Well, we look in different parts of the sky and we pick out stars of the same generic type, different periods in their lifetime, and we put those in serial order. And then we try to explain by a theoretical model of what's going on inside the star, why we've arranged these particular stages in the particular order that we have. So that's what we need to do with planets. And we need to reconstruct the life history of planets. So we start with a protoplanet in the forming of a protoplanetary disk, which can include violent events like the hypothesized collision with Theia, which may have produced the moon. And if we assume that not all terrestrial planets inevitably develop a biosphere, there's going to be one track. There's gravitational wasting that tears down mountains, and the rock cycle. And biospheres evolve over biological timescales, which are driven by natural selection, so they tend to be more rapid than geological timescales. Typical mammal species last 1 to 3 million years. And biological time is nested within geological time, so you tend to see several biological events taking place within a single geological age. And since the Cambrian explosion, we've had basically 3 major divisions of life: old life, Paleozoic, middle life, the Mesozoic, and new life, the Cenozoic. So in the earlier despairing paper over exoplanet atmospheric spectroscopy, the authors asked, what hypothesis or hypotheses is exoplanet biosphere science testing? And well, one of the things we want to test is determining the particularism or the universalism of Earth's development as a habitable planet. And we want to know some of these really basic parameters that we don't know, like how long it takes for life to originate, temporal parameters for the development of life, including the time required to reach a mature biosphere, the temporal parameters of the duration of life, uniqueness or non-uniqueness of major events in the development of life, and the confirmation or disconfirmation of a predictive chemospheric taxonomy. And what I mean by predictive taxonomy is if you're able to do a seriation of the various chemospheric states of planets in different parts of the sky and take planets of the same generic type and see what their typical way of developing is, then if you can identify a planet of that generic type and you can identify its approximate age, you can predict the stages that brought it up to that, and you can also predict the pathways at which it might branch off after that. So we want to reconstruct chemospheric histories, and we will, I expect, find that rapid chemospheric processes imply either life or chemical processes that is a life peer of comparable complexity. And in this context, rapid means a biological scale of time measurement, probably in millions of years. And also in this kind of investigation, whether or not we find life if we find things that are life peers of similar complexity, is going to be of equal scientific interest to study. So eventually, like I said, we want to build a taxonomy where we can say at time zero, when the protoplanet forms, we've got all these kind of different all these different kinds of generic types of protoplanets. Then at the next time forward, we've got various kinds of chemospheres, and we want an exhaustive taxonomy. And then we want to be able to arrange this taxonomy in a distribution that reflects what we find in the universe. So thank you very much to everyone here, and thank you to Sohan and Norcell.